The topic of my talk is basically I'm going to focus a bit more on the data science view related to ADHD diagnostic. In particular, basically, I am interested in performing big data analysis of metabolite data of two particular pathways in order to find out are the differences that we see in children with autism versus the typically developing peers. And ultimately, we want to extend this to also to other children who have non-autism related to developmental delays. Yeah. So if you look at the current definition of autism, that's ultimately what the diagnosis is based on. It's really just by observation. I mean, dealing with basically difficulties in social communication interaction, as well as expression of restricted behaviors and interests. However, when you basically look at some statistics related to ASD, you find that almost everybody has some co-occurring condition that are not, well, they're probably ASD related, but they certainly can't be directly traced to these types of definition for this. Some of the that are frequently co-occurring, you can see is, yes, they have a neural basis as well, but there's also other things like, for example, GI problems, which, I mean, we assume that based upon what other people have talked about here and everything, we know that there's a link somehow, but that's at least contributing, but it's certainly not something that immediately would come to mind when we're dealing with basically neurons and the brain. Yeah, and so this basically forms somewhat the motivation about some of our work to take a bit more of a holistic view here. And what we ultimately want to do is that we want to support the diagnosis as it is, but with some by looking at the biology as well. Yeah, I think everybody can agree that the earlier you can diagnose ASD and can start some form of treatment, this has a better outcome. And there's been a number of papers written on this. I don't need to go into details here. The problem is that this diagnosis, as I just mentioned, it's purely clinical diagnosis. There's no lab test for general diagnostic purposes, but I mean, there are some basic people are working towards at least helping with this in some form or another. But as of now, there's no lab test that you can really use for ASD diagnosis. And the problem we have is if you observe a 2-year-old, it's really hard to determine are they developing normally but slower in some aspects, or is this ultimately, do they have autism, and therefore that's where the developmental delay is. So the result of this, as well as a few other factors, is that ideally we would like to have a diagnosis by 18 to 24 months, but the reality is that in the US, the average is 4 years. In some other countries, it's even later than that. Yeah, so clearly there's a big gap, and anything we could do that allows us to come up with the earlier diagnosis would certainly be very welcome. Yeah, if we have to look a little bit at what do we understand about ASD, I mean, there's been a lot of studies that figure out, so what is exactly, where's this coming from? And I mean, we don't know the exact factors, but clearly they're inherited factors. This is a lot has to do with genes, but also epigenetics to some degree. That makes roughly half of what we know. And approximately the other half are environmental factors that we're looking at. So clearly, there's an interplay between different things that need to be considered for diagnosis. So just having a test just based upon genes is not going to work for other than a very small percentage of cases. Yeah. Secondly, and this is what forms the motivation my work here, we've looked at, so what are the different hypothesized body systems people have looked at? I mean, there's like the gut-brain axis, clearly the immune system plays a role. But two that I'd be focused on was oxidative stress and epigenetics. The epigenetics kind of should cover some of here the commonly inherited part, but there's also environmental factors that play a role there. Well, oxidative stress clearly falls on the environmental side. So we wanted to have at least one foot in each camp and in what we're doing. Yeah. So this brought us to focus on the folate-dependent 1-carbon metabolism and the transsulfuration pathway. These pathways are shown here. I mean, basically, this part that I'm circling here, this is my folate 1-carbon metabolism. Yeah, and it ultimately, one of the results of this metabolism is that basically it regulates DNA methylation, which in turn regulates epigenetics. So that's the link we have to epigenetics. At the same time, the folate 1-carbon metabolism here intersects with the transsulfuration pathway, which leads to the production of glutathione, which is the body's most powerful antioxidant. So if we're dealing with oxidative stress, we would assume that there are some differences that we see here in basically the glutathione metabolism as well. One thing that I want to stress is that oftentimes when people start to look at biomarkers, they usually look at, is there one particular measurement that you have? Maybe it's a metabolite, or maybe it's a gene that I can use as a biomarker. I personally do not think that this is going to be successful for autism. I mean, at least all the efforts people have done show that it's not that simple. There's numerous reasons for it, but one thing you find is that when you're looking at, let's say, metabolite concentration, you have a lot of variability in each group. Yeah? Even in a typically developing group, you can have the values you can have can be all over the place. Same thing when you take measurements from children with autism, you have a lot of heterogeneity in there, and ultimately a lot of this overlaps. So we're probably not going to find one measurement that we're interested in. But given my background, which comes basically from applying data science to biomedical problems, we said, well, there doesn't have to be one particular thing we're measuring. Maybe we see a certain pattern that is that we can link to certain conditions, and that's what we're interested in. So what we've done is that, I mean, we looked at trials that had already been conducted. I mean, part of it is simply because some of these trials take many years to conduct, and if we can make use of the existing data for a start, that certainly would allow us to speed things up. And there was a very nice trial conducted at the University of Arkansas for Medical Sciences, where they had 83 children with autism, 76 children that are typically developing, and they also had some siblings. Now, these are siblings of the children with autism, but they are typically developing. Yeah, and there was a total of 24 measurements, or in some cases, ratios of measurements that were basically analyzed as part of this study. What people have found is, yeah, there are some statistical differences among several of the measurements, but as I mentioned on the previous slide, there's so much heterogeneity. Just based upon the measurement, you cannot tell if the sample came from some of the children with ASD or some of the typically developing. The also nice thing is we had some Vineland adaptive scores, which tells us a little bit about, you can say, there's some measure of severity. We're going to make use of this. So I want to give you some background and how we went about this, and also this tells you a little bit about common pitfalls we want to avoid when analyzing biomarkers. Now, the data I'm using for now, this is made up data for illustration purposes. We're going to go back to the real data, of course, before long. So let's say if I have two measurements I'm taking, this could be one measurement of one metabolite called the X1, and X2 could be a measurement of another metabolite. And then I have two groups. Let's say one of the group could be the children with autism, and another one could be the typically developing. In this case, I said this is made up. I'm going to call one group the red group, the other one the blue. If I want to analyze things based upon the measurement I have just for this metabolite X1, you can find there's a lot of overlap. I mean, this could be even either the blue or the red group. You couldn't tell anything. In fact, even if I try to find statistical differences here, you wouldn't find anything. Statistically, they're not significantly different. If you look at measurements of the metabolite X2, on the other hand, you find, again, there's a lot of overlap here. On average, X2 has a higher value. Yeah, so statistically, you would find a difference between X1 and X2, but you would not be able to determine, based upon just this measurement, which group you belong to, blue or red. But when you look at this, it's clear, like if you draw a dividing line through this, which we mathematically do by projecting this onto a line, then anything to the left of this on this line here will be red, and anything to the right will be blue. So I can actually separate these two very well by using data from both of the measurements. And keep in mind, when I mentioned here X1 as a measurement, there's no statistical difference between the two groups, but at the end, it still gives me very important information combined with other ones to let me do this. As I said, this is a made-up example that we're focusing on with just two measurements, but I can use 3, 4, 5. It just gets a lot harder to illustrate, so I'm not going to do that. The second problem we often run into is, I mean, when we're dealing with patient, now here that I described this as real system, I mean, ultimately, we want to have a model. And this model, for example, should tell us, is the sample most likely coming from somebody who has autism or not? But in order to develop this model, the only way I can access essentially my patient is through data that I'm collecting. And this, you have to take into account there is certain measurement error you're going to get when you collect data. Now, the hope is that if we basically get our model reflects the data as well as possible, that hopefully the model will also reflect the actual patient. Yeah, but what you find is that we only do this indirectly by making this fit good and hoping that this fit here, that there's not too big of an error. But the problem is you can often overfit your data, and that's the problem. You find many cases for these studies. Let me briefly illustrate this again with one example. So if I had true data, you're given in blue by this line and given by this equation. I'm having measurements, these purple dots, which are around the blue data. So this is not bad measurements. If I were to estimate what basically my blue line should look like based upon the data, and in this case, I'm even cheating here. So I know exactly what this model looks like, which in reality, you never know. You just have patient data. But you can see that my black line, yeah, it goes perfectly through all the dots, but clearly the black does not reflect the blue. This is not a good approximation. If I pick something simpler, like this in red, and fit it through the data, yeah, I'm not going to, it won't go through any of the data points, but it gives you a much better approximation. So in short, overfitting is a big problem you have to avoid. And one way to do this is that when we have our samples and we basically have our model, we don't just want to use all the samples to develop the model and look at the error between what does my model predict versus what the sample actually reads. What we do instead is some form of cross-validation where we leave some samples out. So like in our case, I'm leaving the first sample out, I'm building my model based on the other samples, and then I'm predicting what I should have gotten for the sample that was not used for model building, and this gives me an error. I put the sample back in, I move the next one out, and I repeat the same procedure. So by the time I'm done, I have all the errors for each one of the samples are computed based upon something that actually did not make use of the data itself for the model building. And so this gives you some level of robustness. Not as robust as testing it on a completely new data set, but that's always the last step if you basically have enough data. So when we do this and apply it to our folate 1-carbon metabolites, where I said we had 24 measurements, I find actually that I only need to include 7 of these 24. If I go more than 7, my prediction error actually goes down. The fitting error will always actually go up. Sorry, the fitting error will always go down the more you include, but what we want is prediction on data that was not used. So in red here, you see these are the samples from the typically developing participants. In blue are the ones from the ones with the autism diagnosis. And you can see is that there's relatively little overlap actually between the two probability density functions here. Some things of interest, DNA methylation, so our link to epigenetics, as well as the percentage of oxidized glutathione, our link to oxidative stress, were identified as some of the key variables that need to be included for this separation. One thing, I didn't provide the numbers here, but you get with a 96 to 97% sensitivity and specificity. So more specifically, of the 83 children with ASD, we identified 81 correctly, and only 2 were false positives. You know, the 76 children that were typically developing, we got 73 correctly, and only 3 were false negative. One thing as a side note, some people say, well, we don't want to have any false positives. Fair enough. All I've done here in this case, you see this dotted line, that tells me where I put the cutoff. I put the cutoff somewhere to minimize both of these areas where I'm basically making incorrect predictions. You can move this cutoff. Like if I don't want any of the typical developing children from this study identified as having autism, all I have to do is I move this basically down. Yeah, and therefore, yes, my error for missing some of the children in the ASD group will increase, but it will still stay above 90%. So, at least for this sample, so still very, very good. And we've tried to validate this as well on new data. One of the problems is that it's not easy to come by this clinical trial data, especially when we want to have a large number of typical developing children. Yeah, and so what we did here is we made use of data that we had from trials for children with ASD that are ultimately undergoing some procedure or some intervention, but we had some data at baseline before any intervention. And so we had 156 participants, but And we had most of the measurements from before, except we didn't have DNA methylation, and we're also missing measurements for 8-OHD. So what we did is we went back to our original data set, we threw those two out, and we pretended we didn't have this. We built our model based upon what was best and came up with a model that worked best for this case. And then we basically just used it to predict on the new data set. So here is the overlap that you're seeing now for the variable or for the model that's based upon the 22 measurement, or based on the subset of the 22 measurements. You see there's some overlap, and it's bigger because I threw out two important variables. But when I basically then compute my classifier here given by the line and apply this to the new ASD cohort, this is just validation, you see I actually get very high predictive value. Now, I wish I would have data here also for a new typically developing cohort, and that's something that we hope to be pursuing in collaboration with Biopharma. Yeah. So in summary, we basically, the algorithm predicted with high accuracy, both the children had autism as well as the ones that are typically developing. If you want to adjust these type 2 and 1 errors or sensitivity specificity, that is easily to do by just moving the cutoff around. 7 inputs of the original data set worked really well, but if you were not have access to the whole metabolite panel, you may have to adjust that. And we can find that, yes, some of the key inputs, they are linked to oxidative stress and DNA methylation, meaning that was actually a very good idea to focus on this. The second part that I wanted to briefly touch on is saying, well, this was all about, could you tell from a blood sample of somebody, the sample came from somebody with autism or not? But we want to say, is there any data in there that tells us about like some severity score, in this case, the Vineland? And so we have to use a regression model to do that. Now, if you use ordinary regression, it basically tries to map yet your metabolites to the Vineland score. That doesn't work so well here for numerous reasons, in part because we have a lot of metabolite samples compared to the number of children in the study. So you can use principal component regression, which gets rid of unimportant correlations. So that's one option. It's better to use partial least squares, which gets rid of the correlations here, as well as of the correlations in the Vineland. And ultimately, you can use a nonlinear version, which basically uses a nonlinear classification or nonlinear transformation before going through this. And this is kernel partial least squares based upon Gaussian kernels, and that's the work we did. And if you apply this, you find that actually with a model with 5 components, you can actually get a reasonable good prediction of the Vineland score here. This is the predicted, whereas the actually measure one with the R-square of 0.45. I mean, clearly this is not a replacement for the Vineland for these kids, but it shows that there's a correlation there. Yeah. So in summary, I mean, there's a lot of work going on biomarkers for ASD, but as of now, there's no generally accepted one that exists. Yeah, and using data from one study, but then also validating it with data from a second study, we show that the metabolite concentration can be used to determine if basically the sample came from an individual with the autism diagnosis or from a typically developing child. Yeah. Also, there's like in this case, the Vineland, there's a certain correlation between what we find in the metabolites as well as the Vineland score. And so there's significant potential. That's why we filed the IP, and ultimately this IP has been licensed to Biopharma to try to basically turn this into a test that will hopefully generally work. That's work in progress. This has been partially validated, not just in the one study I've shown here, but other research groups have looked at the subset of the measurements that we use and found something similar, but given that it was a subset, their correlations weren't as good as what we had in our study, but clearly it makes the point that this seems to be a promising approach. So in summary, I mean, I would like to thank the members of my research group, my collaborators, funding agencies, and of course you as the audience for listening. And with that, I would be happy to take any questions that you may have. Thank you.
Thanks so much, Juergen. So we've got a couple of questions, and I'm happy to help you on some of them if needed. Number one, how far is this test from being commercially available?
That is really more a question for you, John. I mean, basically, I run a research group at the university. I am not in the business of basically, creating a test myself. That's why we licensed it to Biofarma.
Yeah.
And, John, maybe you want to talk about this, where you stand.
Sure, I'm not the presenter, but yeah, I'm happy to take this one. So we're a few years away from commercialization. So the data that Juergen presented here is obviously very, very promising. And as Juergen said, we did license the technology from RPI. This is something that I've been intimately involved in for a long time, since it originated from Arkansas, where I was even before Biofarma. Basically, where we're at is, in order to get this accepted by the community, we need to have prospective studies moving beyond just retrospective studies. We need to look at multi-center trials as opposed to a single site study in Arkansas. We need to look at things and see how they stack up against the gold standards, such as the Vineland. See how these biomarkers may correlate with other aspects of behavior. Sorry, I think I said Vineland, I meant the ADOS. Looking at how these things correlate with things beyond the Vineland, such as the Mullins and other scales. So we're a few years away, unfortunately, but the stage has been set to do those prospective multi-center trials, which we'll be launching early next year. So if you're interested in the story, tune into Synchrony 2021, and we'll have some data to talk about where we're at and moving that forward. But
thanks, John.
Next question for Juergen. Interesting presentation. It would be interesting if the cases that failed your test were genetic disorders. We imagine cases without T cells in the brain represent cases of genetic conditions more directly affecting brain circuits. So looking at the inclusion-exclusion criteria for the trial, Juergen, if a patient with autism, I guess it ended up being a false negative, would you think that there might be some sort of underlying genetic abnormality that may account for that?
I mean, there's many possibilities. So all I can say is that we looked at all the data available to us, including, there was some patient data collected on basically, if there's anything else that's important to report. Yeah, and it was the few false negatives we had that we could not identify. Yeah, where people incorrectly were classified as typically developing, even though they had autism. Nothing exactly stood out with that regard. Yeah. Now that doesn't mean that when you design new trials that maybe we have to take a broader look at what conditions you want to exclude there or specifically mention with it. But it's certainly nothing that came to mind based on this study. We've done some other studies where we had some genetic information available to us. For the most part, I found that basically did not really improve the prediction capability. Yeah. Now there was a very limited set of genes that we had, so it may be different if more was available. But at least that's what we found from my experience. But I don't want to overgeneralize here. I mean, we're talking about a few other studies where some information was there. Clearly, it's possible that there's more out there that we just didn't have information on that could explain it. But there was nothing that immediately came to mind as far as I'm concerned.
Thanks, Juergen. A couple more questions. Doesn't the metabolite data implicate a systemic mechanism for ASD? In your mind, what do you think about that?
That's a good question, and it's really hard to answer. I mean, what we were focusing on here, and this is why I said we wanted to mainly focus on the diagnostic part, is that we look at, do we see certain patterns in the children with ASD versus different patterns in the children that are typically developing. Now, to what degree this is really more correlated versus causative is not something I can answer just based upon this. Yeah, what we have done, we've done some similar analysis on other studies where basically people ran interventions. And we looked at the analysis of the data at baseline versus the analysis of the data after the intervention. And we found that the metabolites changed, and there was a correlation not only in the change between the changes in the metabolites, as well as the changes in this case also in adaptive behavior from those. So it seems to be contributing, and not, at least at some level, and not just correlating. But I mean, there's obviously many things that can contribute to autism. So I would say, I mean, there are some indications there that it is a contributor at some degree, but I can't go further than that based upon the data and the studies that are there. I mean, there may be more, but it's I just tend to be cautious.
Great. And last question, do you think these metabolites should be measured in the CSF, as they would be more likely to distinguish ASD from neurotypical?
Oh, I think that, now, look, I'm guessing here since I've never seen the metabolites measurement in CSF, but I would assume that most likely would give us, make my job a lot easier to try and distinguish between these two groups. The problem you run into is just that running a clinical trial, getting CSF measurements, especially from typically developing children, is going to be somewhere between very hard and impossible. Yeah, that's why. But if you want my hunch, yes, I would expect that they will likely be, the differences will be even more pronounced there than what we see in the blood. That's a guess, since I've never seen the data, but I wouldn't be surprised if that's what you find. Unfortunately, I think this is a purely hypothetical question, because I honestly don't believe I'll ever see that data as much as I would like to.